Okay, good evening, everybody. Good evening. Welcome to the second night of Brain School during Brain Awareness Week. For those of you who were here last evening, and I recognize a lot of the faces, we got off to a great start, I think. We had a terrific panel, talked about one of the great challenges of our time, drug addiction, particularly the opioid crisis, and got a variety of different perspectives. I think that was a great way to start out and set the tone. We're very fortunate tonight. We're going to have a change of gears about what we'll be talking about. We're very fortunate to have tonight's speaker, Dr. Mike Fox, who's one of our own from the Virginia Tech Carilion Research Institute. Dr. Fox came to us about 5 or so years ago, at the time coming from Virginia Commonwealth University. Prior to that, he had trained at Harvard University, and prior to that, he had done his graduate work also at Virginia Commonwealth University. And prior to that, he'd been an undergraduate at William & Mary, and prior to that, he had started out as an undergraduate at the Military Academy. Mike has had a very successful career. He's known as a world-class researcher himself in understanding how the connections from the eye to the brain develop, and in particular the different molecules and how various things affect that development, and the molecules that guide cells getting to the right place. In recent years, he has extended his research into the realm of beginning to understand how certain nasty things that are out there in the environment can get into our brain and do interesting things. And that indeed is the topic of his presentation this evening, where he will take us through some of the really exciting small things that can have big consequences in our brain. Small things that are normally there to do a good job, and sometimes small things that aren't normally there to not do such a good job. So I'm sure it'll be an exciting evening for everybody. It'll be a little different than last night. Last night we had a panel discussion. This evening it's an individual presentation, but I know Dr. Fox will leave time for questions and be very interested in feedback from the audience. And then I want to also mention last night, the third and final night of Brain School, it's called Kids Night. It doesn't mean it's exclusively for kids. It's also for adults, adults who feel like kids occasionally, or adults who are interested in kids. And we'll be hearing about digital devices and development. Again, a very timely topic with all the devices out there these days, and all of us, including kids, interacting digitally with these things. How do those affect our brain? How do they affect development of the brain? What does it mean for the next generation? We're doing probably the greatest experiment in the history of mankind in terms of what that means, and we'll eventually find out the answer. But we'll try to get some insights on that tomorrow night, and that'll be led again by our own Dr. Stephen LeConte and Dr. Audra Van Wart, and then we'll actually have some students as part of the panel as well. So it should be an exciting evening and wrap up. So without further ado, please join me in welcoming Dr. Mike Fox.
Thank you, Mike, for the introduction. Can you guys all hear me? Can you hear me in the back okay? Yeah. All right. Well, this is a new topic for me to talk on here, so I hope I don't gross you all out by what we're going to talk about. We're going to talk about a few things that can get into our brains and have some really devastating consequences. And so we'll go through 3 parasites. And so I thought I'll just put up the definition here for what parasites are. These are organisms. They come in many different flavors. They live in, on, or off another organism, which we call the host. They benefit from the host. They get nutrients from the host. They get protection from the host. They give very little back to the host. Most times they hurt the host, and so we'll talk a little bit about that today. I thought maybe to start this off and kind of give you a little view, a little glimpse of the power of a parasite, I'd start with a little video. And I'm sorry about the tone of the person's voice, but it's not me, and it's the best one I could find.
Ants are a part of the most disciplined, dedicated social system on Earth. Until that is, they become slaves to behavior-controlling parasites. Suddenly, these rogue ants no longer serve the colony. They're taking their orders from the parasite. Okay, let's back up. Now, how did this happen? It all begins when the ants in Anytown, USA consume the slime of a passing snail. They divide it up and take it back to the colony. This is a blunder of epic proportions. Turns out, The slime is loaded with eggs of a body snatcher called the liver fluke, a type of flatworm. The liver fluke burrows into a part of the ant's brain, and for unknown reasons, it's almost like the fluke enslaves the ants and orders them to carry it to their next hosts. Any grazing mammal host with a nice warm liver will do, but in this case, a cow appears. The liver fluke worms can switch the ant's behavior on and off, causing the infected ants to place themselves in easy-to-eat positions at dusk when mammals are feeding. No cows in sight, ants act normal. Cows appear, ants are in essence ordered to take their positions in purple flowers and latch on. The cows ingest the vegetation, the ants, and the fluke larvae inside the ants all in one bite. Once inside the cow, the worms burrow out of the stomach and into the liver, where they develop into adults and dine on liver tissue. They lay eggs that are excreted from the liver into the bile duct and then defecated by the cows. They don't kill the cows, but the cows become weak and emaciated, devastating herds. And all because of parasitic mind control.
All right, so today we're going to talk about 3 parasites, 2 that do this, and 1 that doesn't, but it's been in the news recently here, and so I thought that it was a good parasite to talk about when we're talking about the brain. And so one of the questions to pose really is, can parasites actually do this to humans? Can they actually influence our minds, our behavior? And I'll tell you the take-home message now, they absolutely can. So the first parasite I want to talk about is Naegleria fowleri. You've all heard of this one before. It was in the news last summer. It's in the news almost every summer somewhere in the United States. This is the brain-eating amoeba. And in the middle panel here is that amoeba. It's a single-celled organism, an amoeba. It lives in fresh water in many of the lakes across the entire United States. But it lives in a relatively dormant state, and it takes temperature increase in the water to actually form this sort of structure when it becomes infectious. The images that flank the single-cell amoeba here are images of the amoeba in brain tissue, and you can see these holes in the brain tissue that have been eaten away by this parasite. So for each of the parasites, we're going to go through kind of like the video and talk about the life cycle of the parasite to figure out how it gets into the brain and why it gets into the brain and what it does when it's in the brain. So in the case of the brain-eating amoeba, as I said, it lives in fresh water. It exists in cold fresh water as a cyst, which is not infectious. Swim all you want in cold water if you like that. Once the water temperature increases, and increases a lot, this perhaps to 90 degrees to 110 degrees is when this amoeba will change from a cyst form into one of these infectious forms. So once it gets to 90 to 110 degrees and you go swimming, this is when you're susceptible to this being infected by this amoeba. So for all of these parasites, an important question is, how does it get into my brain? Because that also will help you stop it from getting into your brain. And so in some of the lectures in previous years, we've talked about how the brain is somewhat walled off from the rest of the body. It's immune privileged. And what I mean by that is there's a barrier between all of the vasculature that fills the brain and all of the what we call neuropil or brain tissue outside of that vasculature. And we call this barrier the blood-brain barrier, or BBB. And it exists in all brain tissue, in all of the coverings up here, the meninges. If you're interested in seeing meninges, we'll have a brain downstairs afterwards. These 2 brains actually have great meninges that you can see. So in most places, a parasite has to break the blood-brain barrier. It has to get into another cell to kind of be a Trojan horse and escape the blood-brain barrier. But in the case of this amoeba, it takes a different route. There's a few places where the blood-brain barrier isn't so good or doesn't exist at all. And one of them is right underneath your olfactory bulb. The olfactory bulb is where neurons that are important for smell reside. They live here in this bulb, and you can see right below them is a very thin bone called the cribriform plate. and there are axons that come through that, and that little hole is where this amoeba comes in. So when you're swimming in warm water in the summer and you take water into your nose, that water that contains the amoeba the amoeba can start eating some of these olfactory sensory neurons here and get into the olfactory bulb. All of the other parasites I'll talk about have mind-altering abilities. I would say this one has a mind-eating ability. The only altercations of your cognitive behavior are because of what's lost, what gets eaten. And in this case, one of the first signs of infection is a loss or a change in smell, and it's because these neurons are being eaten by this amoeba. How bad does it get? Well, if you have this, you develop primary amoebic meningoencephalitis. It's a really big word, and what it means is you'll see encephalitis a lot today. This is brain inflammation. And so this amoeba causes inflammation of the brain and also the meninges, and it's irreversible. There are 3 MRIs here showing different cases of PAM, primary amoebic meningoencephalitis. PAM is a lot easier to say. And you can see the arrows highlight some of the structures where the amoeba has eaten part of the brain. What I want you to take away is not the different structures, but just how varied they are, where they occur, how big they are. Here you have one in the brainstem. Here you have one in the basal ganglia, and back here in primary visual cortex. And here in this view, you can see just how large one of these lesions can be. Lesions that big have devastating consequences. In fact, the survival rate, if you get PAM, is about 3%. So this is an autopsy from someone that had PAM, and you can see just the number, size, and kind of viciousness of these infections.
Mike, can I just add something? We will be selling nose clips for swimmers immediately after the lecture.
So if you go to some thermal pools and you see a sign, you're allowed to swim, but it says don't put your head under, now you know why. So this is the really bad news. Let me give you a little bit more bad news and then some good news. So the little bit more bad news is that the water has to be really warm for this amoeba to thrive and to come out of a cyst into its infectious state. So you can see all of the colored states here are states where infections have been found in the last 50 years or so. You can see we fall in the colored regions. We've had 7 incidents here between 1962 and 2016. The good news is that we've only had 7 in 50 years. We've had 143 cases in the United States during that time, and 139 of them were fatal. So that's good news because it's a really rare thing. It's a really rare parasite. It's a really rare, PAM is a really rare condition. The bad news is it makes it hard to study and figure out how we can prevent it. And the best way to prevent it is to either only swim in ice cold water, wear a nose clip, or don't put your head under the water. As you see the life cycles of these parasites, this typically kills in 7 to 10 days, and it kills so quickly that it's actually not good for the parasite, and the parasite can't propagate, can't spread. And so that's why the incidence is so low. As we get to some of the other parasites, you'll see the life cycle is a circle, much like the fluke worm, the cow, and the ant, where it can kind of keep propagating. And that means those sorts of parasites are going to be much more widespread. So, take home number 1, small things can have catastrophic consequences on the brain. So the second one is Trypanosoma, and I'm not going to say the second word because I'll botch it. But this is a parasite that is single cell, just like the last one. It's an extracellular parasite. It doesn't have to get into another cell. It likes to live in mammalian blood. And you can see this image is an image of blood. All of these round structures are red blood cells. And this is the parasite here. A red blood cell is about 8 microns across. So you can see that these are about 8 to 40 microns in length. They have these tails. They can swim. they're actually coated in a special substance that allows them to slip through the blood-brain barrier and get into the brain. So here's the life cycle of this parasite. This parasite causes a vector-borne disease, which means it can be transmitted by insects. And so here is the culprit, the tsetse fly. Fortunately, we don't have that here. This fly lives mostly in Africa and in South America and a little bit of Latin America. And you can see now kind of the full circle of the parasite's life cycle. And you'll see this for this parasite and a similar one for the next parasite we talk about. So it begins to develop in the fly. The tsetse fly bites a mammal, human, other mammals as well, and deposits, as it sucks some of the blood out, it deposits eggs of this parasite, which then propagate through the blood. And you'll see there's not really a picture of a brain here. So at first, this parasite doesn't get into your brain. This parasite just likes to live in blood, and the next time another fly comes and also takes some of your blood, it takes some of the parasite, and it keeps this cycle going. As long as you're willing to give blood to a tsetse fly, the parasite can live happily getting nutrients from your blood each time this cycle happens. So what becomes really interesting is when eventually this parasite also makes its way into your brain. So what's fascinating to me about this parasite is it targets one place in your brain. It has a mechanism where it could get anywhere in your brain. This substance on its surface would allow it to get to any part of your brain that it wanted. It could slip through any part of the blood-brain barrier, but it seems to only go to one region in the hypothalamus. And so here's a sagittal view of the brain. Here's the eyeball. And right here is the hypothalamus. And if you zoom in, it's this area right here. And it seems to target one region of the hypothalamus in general. And I should say, sorry, the hypothalamus is a really important brain structure that does a lot of regulatory functions, autonomic functions, and things like that. And one of the nuclei within the hypothalamus is called the suprachiasmatic nucleus. And it sits right here. It's only about 50,000 neurons in your brains. And what's really important about it is this really small nucleus with a really small number of cells is the master regulator of your circadian clock. So right now, the sun is out, I hope. It's still cold, but I think the sun is out. And you're awake. When the sun goes down, you get tired, and this is your cycle. If you get on a plane and you fly to Europe, you feel jet-lagged for a few days, it's this little part of your brain that's trying to reset your circadian clock and adjust your internal clock to the change in light in your environment. So it turns out that there are very specific neurons that live in the suprachiasmatic nucleus, which contain a specific type of neurotransmitter, and they project a set of axons or make connections eventually with the pineal gland. And the pineal gland contributes to your sleep-wake cycle by producing melatonin. So this entire kind of axis here is important for that sleep-wake cycle. What's fascinating is that this little parasite only affects the suprachiasmatic nucleus. It destroys those neurotransmitter systems, and that not only destroys the suprachiasmatic nucleus, but changes melatonin expression by the pineal gland. And this is why it's called sleeping sickness. Most times when you get sick, you get tired. You sleep all the time. If you had the flu, you'd be very tired. You probably wouldn't want to get up. And this is a very different disease. You don't just get tired. You get tired during the day, and you're awake at night. It flips your circadian system. And there's an advantage to that, to the parasite. Why would a parasite, a vector that lives in a fly, want to do that? Well, the truth is, we don't know. But what people hypothesize is if you're asleep during the day, when a fly is awake, it's a lot easier for it to target you and to eat and not get swatted than if you were active. And so the hypothesis is that this parasite has evolved to take control of your sleep-wake cycle. And by changing it, it increases its fitness. And when we talk about parasites, increasing fitness means that it can survive, that it can spread more efficiently. And so this is how this parasite does it. And so this is going to start a series of parts of this lecture where we're going to hear about neurotransmitters. And so I want to highlight that now. As we go through some of these parasites, we're going to talk about neurotransmitters. Neurotransmitters are also small things that have really big consequences in our brain. And so this was just to remind me to say that keeping this cycle going requires the tsetse fly to be able to have access to a human. And the less active that human is, the higher the fitness of this life cycle. So small things like this parasite not only have devastating consequences to our brains, but change our behavior in order to favor their survival. Okay, I don't work on any of those parasites. The next parasite I work on, and it's my favorite. And this one is really scary. So this little monster is the Felis silvestris catus. It's not that scary. And at this moment, you're probably thinking, I never knew that's what a cat was called. That's what a cat is called. And you're probably thinking, well, that makes a lot more sense where these guys got their name. Sylvester the cat was named after the Latin name of the domestic cat, as was Felix the cat. Okay, so it's not this guy that's so scary. I hope that's not the only thing you remember from tonight. This guy isn't so scary like this. He's really more scary like this. All right, so the cat itself, not a parasite, but when it's in this position, it's primed to release parasites. And the parasite that it releases is called Toxoplasma gondii. And this is a little different than the other parasites I've talked about. This is an obligate intracellular parasite, which means that it wants to live inside the cell of a host, not in the extracellular environment, not in the blood. It wants to actually get into cells and reside in there. And in its most virulent, active form, it looks like these tiny little crescents. Now, it's smaller than all those other parasites I talked about because it has to live inside of a cell, but it is just as effective as taking over control of an organism's brain. Okay, so let's go over the life cycle of this really cool, not scary parasite. Kind of scary. All right, so the only place it can sexually reproduce is in the cat intestine. So the cat is its definitive host. And so we're going to start this life cycle right here, where this parasite is in the cat intestine. It can get shed from the cat in feces, which is hence the litter box image. And once it's shed, it is in an oocyte form that's quite stable, and it can live like this for a while. And once it's shed, it eventually gets picked up by a foraging animal. Foraging animals might not live in your litter boxes if you have cats, but of course this parasite evolved pre-litter box era. So what do I mean by a foraging animal? It could be a lot of animals here. You see a mouse, you see a rabbit, you see a chicken. There's others. Pigs are also foraging animals. Goats are foraging animals. So any sort of foraging animal will likely pick up some of the dirt and feces that contain these oocysts. Once that oocyst gets into the digestive tract of a secondary host or an intermediate host, it begins to differentiate into that virulent form that I showed you a picture of, which is called a tachyzoite. The tachyzoite then invades the cells within the gut, and it replicates. And when enough have been replicated, it will come out of those cells, get into the bloodstream, and spread throughout the organism. Now, once it does that, the animal is under an active infection. There's probably some consequences of that, much like getting a cold or even a flu. If we picked it up, we would have the same sort of immediate response. But eventually, this active form transitions into a more chronic form where there isn't an active infection. And that requires the parasite to find just a few tissues where it likes to live. One of those tissues is skeletal muscle. So any skeletal muscle will work for the Toxoplasma gondii. And the other is brain. I wouldn't be talking about it if it didn't like to reside in brain. So let me tell you how that happens. So for the other parasites, it either came in through the nose, and the olfactory bulb, or it could actually slip through the blood-brain barrier. In this case, there's three ways which we think Toxoplasma gondii can get into your brain. One is that Trojan horse mechanism I talked about. So it can get into other types of cells, like macrophages, and then they can cross through in an active manner the blood-brain barrier. That's mechanism number one. The second is that an extracellular parasite can slip through the blood-brain barrier at tight junctions, not through the cells here, but just through the tight junctions, the connections between those endothelial cells. And the third way is to actually infect the endothelial cell, which is the innermost wrapping of the blood vessel, replicate in that endothelial cell, and then shed new tachyocytes into the surrounding area. All three of these ways are possible for toxo to get into the brain. Okay, so now it's in the brain. If the animal dies, toxo doesn't get back into the cat intestine, and that's not good for toxo. And I'm sorry, I keep saying toxo. That's what we say in the lab. Toxo equals Toxoplasma gondii. It's just a lot easier to say toxo than all that other stuff. So if the animal dies with this in its brain or its muscles, it's not very useful. So what does it do? How does it get back into the cat intestine? So I want to talk about this guy first. So this is a mouse. This is a common house mouse. I could have picked a chicken, I could have picked a pig, but this guy was cute, so I picked the mouse to talk about. What do mice do? They like to forage. They'll take dirt, but maybe cheese would be better. And they're pretty skittish. They like dark spaces. And what do they really not like? Cats. They're extremely terrified of cats. So if this guy were in an environment where it smelled cats, where it smelled an actual cat, or where it smelled cat urine, it would get out of Dodge quick because it doesn't want to get eaten. What Toxoplasma gondii does is it changes that behavior. It makes the mouse like the smell of cat urine. Now, the mouse plays in cat urine while the cat gets ready to eat the mouse. And for the parasite, that's great news. Really bad news for the mouse, great news for the parasite, because now the parasite gets back where it wants to be, which is in here in the cat intestine. And here is the life cycle of the Toxoplasma gondii. All right? So what I want to do next is talk about two parts of that life cycle that we talked about briefly. The first is an active infection. I want to talk about how that relates to humans. It causes a disease called Toxoplasmosis, and that is the active infection is caused by these tachyocytes. Once we do that, we'll come back and we'll talk about what a chronic infection of Toxoplasma does. And this is a cyst which contains lots of those tachyocytes, and the cyst is called a bradiocyte. Okay, so I've kind of summarized here in a smaller form that life cycle that I talked about. Starts with cat. Cat releases oocytes in fecal material. Foraging animals pick it up, gets in their muscle, changes their behavior, eaten by cat. Nice little life cycle here. Unfortunately, we can come into that life cycle in a few different ways. The one that everyone asks me about is cat litter. Yes, you could get Toxoplasma gondii from cat litter. It's why anyone who becomes pregnant is told they should not change cat litter anymore. The reality is most people don't like to play in cat litter. Please don't tell me if you like to do that. And most people probably wash their hands after they're done doing whatever they have to do in the cat litter. So yes, you can get it from here, but it's actually a really minor way to get Toxoplasma gondii. You could get it from working in dirt. Of course, dirt is where the feces would be if there wasn't a cat box. And so that's actually a really common place to get it. You can get it from any sort of fruit or vegetable that hasn't been washed. Same reason, touches dirt. Dirt might contain Toxoplasma gondii. But really the reason is we humans have evolved to really like the taste of some foraging animals. In the South, I think we particularly like this one. And this one's muscle contains Toxoplasma gondii because it also is a foraging animal. So these are the sources through which humans come into contact with toxo and how we can get toxo. Now, as I said earlier, if I get toxo, I get a little bit of a cold, or I feel like I have a cold, or I feel like I have a flu, but it passes in just a few days, and I don't feel any worse for the wear. Toxoplasma infection really affects just a small subset of the population. One I mentioned are pregnant mothers, and again, it doesn't really hurt the pregnant mother, it hurts the fetus, and that's called fetal toxoplasmosis. And the other is any population that's immunocompromised. So someone who's taking medication because they've had a transplant, so they're taking immunosuppressants, someone with HIV or AIDS. These are populations that can get toxo and where it really causes a devastating disease. And in both cases, either the fetus or the immunocompromised person, toxoplasmosis causes a very similar thing to PAM, maybe not so bad, but it's encephalitis, so swelling of the brain and an immune reaction in the brain, which is bad for neurons. So while it's not the most common disease you've ever heard of, it is actually the second leading cause of death related to foodborne illness. And I think this drives home the point that this is where we're getting toxo from, not the litter pan that's covered up, and that it's a significant threat to human health. Toxoplasma gondii also, during development, is classified in a torch disease. And the word torch here really was used to define this kind of inflammation of the brain, like a fire in the brain. And so they also conveniently were able to match up many of the diseases that cause this in the developing fetus to spell out torch, except for the other, which kind of just got everything that didn't fit into the word torch. So what is a torch infection? A torch infection causes mild maternal illness, but severe, potentially fatal fetal illness. And if you treat maternal infection, it really has little to no impact on the fetal outcome. And this is why these are devastating diseases. And I'm not going to talk much about it today, but you've heard about one of these in the last 2 years in the news. A new virus has emerged. It's not really that new, but it's new to humans, and it's called Zika virus. Everybody's heard of Zika virus, probably. So Zika virus gets classified as a torch disease now. And the consequences of Zika infection in a mother to the fetus are almost identical to that of Toxoplasma gondii. So the images you see in the news of children with microcephaly or small brains is something that you would see with a toxo infection as well. So Zika virus is another small thing with really big consequences in the brain. Okay, so now I want to move to this guy. This is the cyst. This is the chronic infection. Once you are infected and the tachyocyte moves from this active infection and finds cells that it likes to live in, like muscle cells and brain cells, it replicates, but it stays within this kind of glycomatrix, kind of a wall that it creates inside the cell, and it can grow quite large. And so I want to show you an image of this that we captured here at VTCRI, and the student who did this is somewhere back there. So this is a side view, a sagittal view of a mouse brain. And if you were to cut the mouse brain like this, you would see a section like this. And in green, you can kind of see the outline of the brain. And this outer part here is the mouse cerebral cortex. When we go downstairs and look at the brain, this is the mouse's version of all those bumps and grooves that you see on the side of the brain. And if you were to zoom into this area and use electron microscopy, you would see something like this. I know it's hard to figure out what it is, but I can walk you through some of it. These weird structures, these are blood vessels. All of the lighter circles are cell bodies of neurons, glial cells, other cells that reside in the brain. You can see lots of them. All of the gray material between those cell bodies are all the wires of the brain. And right here is a Toxoplasma gondii cyst. So let's zoom in so you can see that. Here is an image of that exact cyst. And again, it's a little difficult if you haven't spent years and years looking at these images to know what the heck anything is. So we've pseudo-colored them for you. And so what you can see in yellow, if I get my colors right, in yellow are neurons. And so here's a neuron cell body. Here's one of its dendrites coming off, another neuron, neuron. In red are a type of glial cell, kind of an immune glial cell called a microglial cell. And in blue, the biggest thing in the whole picture is a Toxoplasma gondii cyst. Now, this is residing in a neuron. So look at the size of a neuron, and look at the size of the Toxoplasma gondii cyst, which is inside a neuron. This is like if I swallowed a cow and tried to give this lecture. This neuron is actually healthy, and that's important for the cyst. The cyst wants to keep itself alive. It doesn't want to kill its host. So why does it hide in a neuron might be one question you have. Well, it turns out neurons are really bad at making signals which might trigger an immune response. So if you're going to hide in a cell, you should pick a cell that can't say, "Hey, the cyst is in me. The cyst is in me." And so that's why Toxo likes to hide in these neurons. Now, once it's in there, it actually uses some of the resources of that cell to survive. So it kind of eats up some of the inside of the cell, some of the things the cell makes, but it also starts to take over the cell to control things like behavior. And it does that by taking over some of the neurotransmitter systems. So we think it takes over some of the dopamine systems to control some of the fear, the loss of fear in rodents. My lab has shown that it takes over some of the inhibitory neurotransmitters like GABA, and other labs have shown that it takes over some of the glutamate or excitatory neurotransmitter systems. So it hijacks all of these systems so it can stay alive, so it can hide, and so it can control an animal's behavior. Okay, an important part. Once you're infected with Toxo, you're always infected with Toxo. You might not have an active infection, but you already have it. All right, so now here's the scary part. I want you all to look to your right and to your left and see how many people there are in your row. So we got 6 people here, about 8 people there. So 1.5 of you here have it, 2 people in that row have it. That's how prominent it is in the United States. 20 to 30% of the US population have Toxoplasma gondii in their brains. That's actually a low percentage compared to other countries. Remember how I said you could get it? One of the ways is through food. And countries where they prefer the taste of food to how well food is cooked, their rate goes up. So if you're in France, 50% of the population is Toxoplasma gondii positive. So yes, lots of us have it. So and I can actually probably ask you a bunch of questions and make an educated guess whether you fall into this. One of the questions that is kind of almost diagnosable is if you grew up on a farm, this number probably is close to 100%. So I usually give these talks to scientists, and they're like, well, it's different. It's a rat or a mouse. We're different. The parasite has evolved to take over the mouse brain. There's no reason for a parasite to control our brain. And I didn't really have a good argument for this until 2016, when a group studying chimpanzees basically replicated the mouse experiments with chimpanzees. So I should say a house cat isn't a predator of a chimpanzee, but a leopard is. And a leopard can also be a primary host of Toxoplasma. So the same circle can exist. Just take out the house cat, put in the leopard, take out the mouse, put in the chimpanzee. And sure enough, this group showed that infected chimpanzees not only aren't fearful of the smell of leopards or leopard urine, but they're actually attracted to it. So what you can see here is they took urine. Can you imagine if I had a graduate student here do this? Go collect urine from a human, from a leopard, from a lion. Would you let me do that? I don't think so. Okay, so some poor graduate student had to collect urine from these 3 and then test how a chimpanzee responded to it, whether it was attracted to it, whether it would investigate it, or whether it would pull back from it. And sure enough, the human pee really had no effect on chimpanzee behavior. It didn't matter if the chimpanzee was uninfected or infected with Toxoplasma gondii, but when they used the leopard urine, they saw that the investigations of the control, the non-infected, was greatly reduced, and the investigations by the infected rose dramatically. So not only was it not fearful of it, but it was actually investigating the leopard urine. Really bad if you're a chimpanzee. And so this idea that Toxoplasma controls just a rodent brain is probably not true. So 20 to 30% of our population has this. What does it do to us? Well, it alters our behavior. There's all sorts of behavioral studies that have shown that Toxoplasma-infected individuals make riskier choices. They don't look both ways before they cross the street. Not a joke, that's a study. They are more inclined to do adventure sports, skydive, not a joke, that is a study. They have less fear, they have more aggression, there are other behavioral traits. It's not everyone that's infected, but it predisposes you to be more likely to make those choices, to make those decisions. My own lab became interested in it because it also is associated with a higher risk of developing all sorts of psychiatric illness. And the one we were most interested in is schizophrenia. Turns out, if you are recently infected with Toxoplasma gondii, you're more likely to develop schizophrenia than any single gene mutation we've identified to date. And I don't mean we as my lab, I mean we as in the world. So I think that's really significant, and that's why we're interested in studying what schizophrenia does to neural circuits in the brain. All right, so small things, Toxoplasma gondii, can have mind-altering influence, again, taking over our neurotransmitter systems. And there's other things. I could go on and on and on. I have you here for another 2 hours, so we'll go through all of these. We could have gone through viruses. We could have talked more about Zika. We don't know that much about what Zika does to the adult brain yet, but I'm sure those studies are ongoing. We could talk about rabies, where there's a huge literature about how this actually takes over our brain. There's something called hydrophobia in rabies, where you're scared of water when the rabies virus actually makes it to your brain. And there's all sorts of reasons why we think that's important for the propagation of the virus to the next host. We could have done this with toxins and talked about alpha-latrotoxin from black widows or other types of spiders, snakes, bacteria. You kind of got a little slice of this yesterday, I think, because drugs are small things that have really devastating consequences in the brain. So as I was putting all this together, I really focused in on parasites because of the new interest in my lab, but I got one question that kept coming up. And when I said I was going to talk about parasites, people kept asking me, oh, you're going to talk about Brain on Fire. And so this, how many people have seen this story, have seen the movie? Not many? Okay, so good. So apparently 3 people did here because they asked me about it. So this is a journalist, a young journalist, who several years ago seemed to be a very healthy individual. And then all of a sudden, she developed all sorts of psychiatric illnesses. She had very violent episodes. She began experiencing delusions. She had an entire month of her memory was gone, during which time she had delusions and violent episodes, and she had no memory of it. They had a huge trouble diagnosing her. They eventually diagnosed her with schizoaffective disorder. It turned out not to be what she had. And so when she got healthy again, fortunately, she wrote about this, kind of used her skills as a journalist to write this amazing book, which she called Brain on Fire. So what causes this? What takes a normal individual and in the flip of a switch causes all of these problems? Could have been a parasite, could have been any of the things I just mentioned. Turned out not a parasite, much smaller than a single cell parasite. Turned out it wasn't a virus, much smaller than a virus. In this case, in this individual, and we actually had one of the scientists that worked on this in the very early stages. I should say this disease was first diagnosed in 2007. In 2013, we had Rita Belize-Gordon here give a talk, and she was one of the people that first identified what this condition was. It turns out to be an autoimmune disease, where the patient themselves was creating antibodies that were targeting proteins important for synaptic function. And so here's just an image of an antibody. These are antibodies or protein structures that are generated by B cells in your immune system, and they're important for your immune response. At the end of this Y-shaped structure, you can see what we call antigen binding sites. Those are targeted parts of this protein which only bind to one other thing. We call that the antigen. And in this case, unfortunately for the writer, the antigen was something that was important for brain function. So why did she develop these antibodies? Well, it turns out she had a tumor, not in her brain, and that tumor was erroneously making these proteins that are important for the brain. And so her immune system saw them, tried to target them, made antibodies against them. That was probably good for the tumor, but it ended up being really bad for the brain. There are actually many of these sorts of autoimmune diseases that are caused by these tumors, and because it's a tumor-related condition, but not to the tumor itself, we call these paraneoplastic conditions. So for her, it turned out that this antibody was directed against something at a synapse, absolutely critical for brain function. So this is a very brief, small depiction of a generic synapse. This is actually the synapse here. It's the connection between two neurons. It's how the neurons communicate. It's actually a gap. It only works in one direction. We say it's unidirectional. So the neuron that's before the synapse we call presynaptic, and the one after is postsynaptic. And in the presynaptic neuron, you can see all these little green dots. These are neurotransmitters. They're chemicals that the presynaptic neuron is going to release to activate the postsynaptic cell. This is chemical neurotransmission. You can see they come down in these vesicles, they get released, and then they bind to something on this postsynaptic cell. Those are called neurotransmitter receptors. It turned out brain on fire was caused by antibodies against one type of neurotransmitter receptor called an NMDA receptor. All right, so the antibody bound to the NMDA receptor, it inactivated it, and it actually caused it to be taken off of the membrane. And so this caused all of those conditions. Fortunately, once they take the tumor away and give a few drugs, they can actually reverse this. And so she was able to regain her normal life, write a best-selling book, have a movie about it. So I think there's some good news about these, since I've given you lots of bad news about brain-eating parasites today. This is not the only type of this antibody-mediated encephalitis. There are many. There's other types where antibodies target other types of receptors, such as myasthenia gravis. Myasthenia gravis is an autoimmune disease where antibodies are generated against a different neurotransmitter receptor. Some against other parts of the synapse, which are really important. So the synapse has all of these channels which allow ions to flow in and out of the cell. And there are some of these antibodies are generated against these channels, or enzymes that break down neurotransmitters, or other types of molecules that are important in the synapse. A lot of them are targeted towards the synapse, and a lot of them have these mind-altering possibilities. So, on top of all these parasites and viruses, there's even small molecules that your immune system make that can have consequences on brain function and behavior. So really, the take-home message here is that small things, really small things, have really big, really big consequences on the brain. And in many cases, the reason for this is they target neurotransmitter signaling. Neurotransmitters are highly conserved between species, so it's very advantageous for parasites and other things to take advantage of them. So I'm preparing this lecture. I came up with this kind of silly, small things, big consequences. I kept saying it to myself, and I was like, you know what's a small thing that has really big consequences? Neurotransmitters are small things that have really big consequences, and that's why parasites take advantage of them. Neurotransmitters are single molecules typically, or very small peptides, and yet they have all of these amazing roles in synaptic function. Here's just a list of just a number of these neurotransmitters. Some are just simple amino acids. Some are small strands of peptides, just a few amino acids in length, really small, much smaller than anything that we've talked about today. And yet they're important for all of our brain function, all of our ability to move muscles, to have thoughts, to have memories. And so these are truly small things with really big consequences. Now, as someone who likes to talk about this stuff, anytime there's an article about something like this in a kind of a public setting, I get excited. I like to read it. One of my favorite journals is actually National Geographic. They've had just a set of amazing articles on brain over the last 15 years or so. And they had one where they talked about neurotransmitters and synapses and how they're small things that have really big consequences. And I was really excited. And this was many years ago. Unfortunately, I read the next issue as well, and in the comments someone wrote, I was disappointed by what's in your mind with a few keystrokes. The author abolished the human spirit. it's hardly possible that the great civilization that your publication explores were created by the firing of synapses. So at first, that was a little disheartening. And you're probably wondering, like, this happened 4 years ago, and yes, it's true that I photographed it, and it's been in my phone since, and I delete pictures of my kids before I delete this quote, because I find it inspiring, and it's true. For those people that aren't in our world, if you're not a neuroscientist, if you're not in the nuts and bolts of a brain all the time, it's really an overwhelming thought that neurotransmitters, these small things, can have such big consequences. And in fact, if you went back 100 years, neuroscientists would have said the exact thing. And in fact, they did. So at the turn of the century, 100 years ago or so, chemical neurotransmission was fought by almost every neurobiologist. Neurobiologists that went on to win the Nobel Prize, the highest award you can get in science, fought against the idea that chemicals, that these small chemicals could actually control brain function. But it turned out to be true. And I want to share with you briefly the experiment that made it happen. And this is really just to pay respect to all of the cardiovascular scientists here, because they hear a lot about the brain. And so I want to show one experiment where we learned something about the brain by studying the heart. So this was an experiment done in 1921, where they took a heart, and the heart is innervated by the autonomic nervous system, specifically by the vagus nerve, which is carrying parasympathetic input. Now, if you know anything about the autonomic nervous system, it's two halves, the parasympathetic and the sympathetic system. The sympathetic system is your fight or flight response. The parasympathetic system is your rest and digest response. So with just that little bit of knowledge, if I told you that the parasympathetic system was innervating the heart, and if you stimulate it, what would happen? So here's what happens. Here's your heartbeat. And this is just measuring contraction force. And then you stimulate the parasympathetic input, rest and digest. You're going to slow your heart rate. You're going to drop the amplitude of the contractions and come back. What these, at the time, really brilliant experimenters did is they coupled the bath that this heart was in with another heart. And they hypothesized that if it was a chemical that was doing this, when they triggered the synapse at one heart, the molecules would eventually make their way to the second heart and cause the same exact response. And that's what you see here. You can see it's delayed from the onset of the stimulation, but you get the same slowing of the contraction rate and amplitude of the heart. And this showed that there were chemicals that could do this. And they later went on to show that this chemical was called acetylcholine. They did this in 1921, and they won the Nobel Prize for it in 1936. So this is a little easier to imagine, just contraction of muscle. But could a neurotransmitter really be important for brain function? For all of the amazing things we do every day? For all of the amazing things that National Geographic reports on that we're all in awe of? And to me, when I was putting this together, I thought, well, you know what? Parasites kind of show us that that's the case. And I would posit that how parasites or even viruses or even antibodies take advantage of our neurotransmitter systems to control our behavior really highlight how small things like neurotransmitters can have huge consequences on our mind, on our behavior, and on our brain. And with that, I'll be happy to take a few questions before I dart downstairs to show you all the brain.
Thank you, Mike. We've got time for some questions, comments. Floor is open. Way in the back. Please stand up so everybody can hear you.
What about parasite that's in raccoon poop?
Raccoon poop parasites, Mike.
Okay, so I thought I'd preface this. I'm a neuroscientist that has just gotten into studying parasites. If you go to the CDC website, which I just did for the first time 2 weeks ago, there are hundreds of parasites. I have no idea what parasite lives in raccoon poop, although I do know that they probably also can have toxo as well. It's a little broader than just cats, but they probably have toxo as well. Do you know the name of it?
I don't remember it, but I know that it's incurable and it causes risk-taking behavior.
Sounds like toxo.
Yes, in the front. Please stand up so everybody can hear your question.
If you're already, if some people are already toxin positive, what kind of
So this is a really good question. What if you're chronically infected with toxo and become one of those conditions where an active infection is catastrophic, pregnant, immunocompromised? And in most cases, if you have a chronic infection, it won't affect you if you have a baby or if you become immunocompromised. What you need to do is have another active infection. There are some places, so that I skip over. There are actually different strains of Toxoplasma gondii. There's 3, maybe 4 strains. And the one we have in the US it's good. It's not good, but it's better than the others. So be happy about that. Don't go to Brazil. And this type is, it's very unlikely to have a chronic infection triggered back into an active infection. In other strains, it actually happens, but only in a few places. And actually, it's the reason that I started studying toxo, because one of the places is the retina. And the retina, oftentimes, it has a blood-brain barrier. We call it a blood-retina barrier. I don't know why. It's the same thing. But it can break a lot more often than the blood-brain barrier, and so then you can have infections in the eye. It doesn't happen with the US strain of Toxoplasma gondii, but it does happen in many of the strains that are in Brazil. And that's just one small part of the brain where you can have a chronic infection turn into an active infection. But in most cases, for us, it doesn't happen, so the risk is really, really, really small.
Right there.
I'm wondering if inherited psychiatric illnesses are inherited because of genetics, or is it an inherited defect in the neurotransmitters?
So are neuropsychiatric disorders, are they genetic because of inherited genes or because of disrupted neurotransmitter signaling? Yes.
Doing that.
Yes. Yeah. So those go hand in hand. As I alluded to, the truth is that with many of these neuropsychiatric diseases like schizophrenia, they're probably multigenic, meaning many genes can be affected and lead to them. And we know very few genes where if I saw that you had a mutation in that gene, I would know that you would develop that syndrome or disease. And so they can be associated, but it doesn't mean it's causative yet. And some of them are in neurotransmitter systems as well.
Mike, before the next question, I just follow up on his question. So you raised the specter of psychiatric disease as a contagious disease, at least in some cases. So we just heard about that. So what are the possibilities and what evidence is out there with respect to other psychiatric diseases, other infectious agents? Kind of like when the world of medicine was revolutionized, when people realized ulcers were an infectious disease, and nobody believed it, and used to throw tomatoes at scientists and doctors who said that at meetings. And now in the literature, people are finally starting to say a little louder, maybe some psychiatric diseases are contagious. How common might that be?
Well, I think what they certainly are is that immune systems can trigger them. So which we didn't know many years ago. Many conditions can be exacerbated or caused by these sorts of inflammation events that you see. Some of these, so that's a really big area in the field now, trying to understand that balance between the circuits, between infectious disease. A lot of the effects that you see from Toxoplasma gondii might not be specific to toxo. They might be specific to many diseases where you have an infectious component. And that infectious component might be from something that is contagious, something that can...
General brain inflammation.
Yeah.
Anything.
Yeah, so a lot of times when you have something that causes brain inflammation, because we were talking about mind altering, I didn't get into every phenotype, but one of the phenotypes that is a hallmark of many diseases where you have brain inflammation are seizures. And I think they're probably less specific to, say, Toxoplasma gondii, where you can have seizures, or malaria, or other types of things that cause brain inflammation. So I think brain inflammation in general, it causes many symptoms associated with these sorts of diseases. And those can then change circuits, change neurotransmitter systems, and lead to kind of irreparable changes in how our brain works.
Yes, you mentioned genetic changes. Could that possibly include chromosomal translocations?
So the question is, could parasites change the genetics of an individual?
And cause the chromosomes to...
Yeah. I don't know if anybody has looked at that. I do know there are many groups that are interested to know whether parasites cause epigenetic changes, which is a slightly smaller event than true chromosomal rearrangements. But whether or not being infected might make you more or less able to read certain genes and to transcribe them. There are many groups looking at that. I don't know if we really have any targets yet that have been identified.
Yes, right there.
What is the relationship between vitamin D or vitamin D3 and the improvement in depression?
I have no idea, and I'm not going to try to wiggle my way out of that one.
Good question for a psychiatrist.
Yeah, where's Bob Tressman?
Thanks. So a lot of the behavioral effects you talked about related to toxo, it seems like those maybe are coming from like associational studies where you're looking at two different populations. Wondering how well characterized the mechanisms by which those behavioral changes come about are at this stage.
So the question is, how concrete do we know how behavioral changes in humans, not just in rodents, results from Toxoplasma gondii? We have no idea. So many of us are trying to use rodent models to take a single, say, behavior and try and pin down a neural mechanism. But we're really at the early days of this. A lot of people actually study Toxoplasma gondii in neuroscience, but they've studied it from purely a model system, that this is a parasite that in the scheme of things, it's pretty easy to get your hands on. It's not very contagious as long as you don't eat the mouse. And so you can use it as a tool to cause immediate brain inflammation. So lots of people has used it for that. We have just a wealth of knowledge of what toxo does in the first 7 days. We have much less knowledge about what Toxoplasma, chronic infections of Toxoplasma does, and how we can take individual behaviors and figure out each underlying mechanism. It's one of the things we're really interested in, and I think it's an exciting field.
Mike, if I might ask, my wife who's in the audience orders her hamburgers and steaks medium well these days. And I think that was with respect to fear of mad cow disease. But with respect to foragers like pigs, for those who eat pork, you want to say something about the denaturing of protein and what we eat or acid or anything to make people feel better?
I'll simplify this. 145 degrees kills a toxocyst. 145 degrees. So there are bacteria, other parasites which are way more resistant. It actually is pretty easy to kill. I mean, that's a pretty low bar to set, 145 degrees. We're not saying 160 or anything like that. 145 degrees kills Toxoplasma in pork and everything like that. It could still be a little pink.
Yes, right here.
Just on the major subject, as a layman, when I was taking away small things and big consequences was if I wash my fruit and vegetables and I wash my hands and I cook my pork, I don't have to have too much worry about it.
Correct. Yeah. I just saw. Yeah. Don't swim in warm water. I just saw an article this week, and I'm still kind of new to Twitter, but I tweeted it out, that foxes can carry Toxoplasma gondii, and that in neighborhoods they're finding it that it can be shed in gardens. And so even in your backyard garden, you should wash your vegetables, even though you're growing them back there and maybe you're not using any chemicals or anything like that. Even in that environment, you don't know what's happening there in the middle of the night when all these nocturnals are out, animals are out. Wash your vegetables even in your own garden. There was another question here.
If
schizophrenia or something like that, then will that one day maybe prove that all the pharmacological stuff they're giving people with schizophrenia really don't apply because it's being affected by a parasite? And
Well, so I would say, I would never say toxo causes schizophrenia. It probably makes you more susceptible to it. But I think all of the pharmacological treatments would still work because whether you have a genetic disorder which causes schizophrenia or whether you have this parasite that causes it, we think that in the brain it's still the same thing. It's still taking over certain circuits, certain neurotransmitter systems. And so even though it might have different causes from an external point of view, solving it might take the same approach at a cellular or circuit level within the brain. So I think it shouldn't be like either or that because toxo is more highly associated with it, it is a more important one. I think they're all working together, and many of these things lead to these sorts of neuropsychiatric disorders, which is really why they're very difficult to treat and why they're very difficult to understand even when you use model organisms and you're trying to kind of figure out what is the mechanism underlying them. So I don't think any of this would cause us to go find a different way to treat or to study neuropsychiatric disorders.
So Susie, maybe if I can add another perspective. I think saying the treatments, pharmacological treatments are treating symptoms. They're not curing the disease. So the root cause, as Mike talked about, whether it's inflammation, what you're doing with probably what most of the drugs that you're thinking about that you would know about is directing it at symptoms or manifestations. You're not going back to what the root cause was, basically. Yes, sir. Stand up, please.
Well, but you could imagine that you present with psychiatric symptoms and the first standard course of treatment is antibiotics.
Well, I think
It's not yet.
I think every case is probably different. Like if you take brain on fire, maybe had the same symptoms as someone who developed schizophrenia from a toxo infection, right? And yet they're very different and they have to be treated differently and diagnosed differently. And that was the problem with brain on fire is they tried a lot of standard treatments and until 2007, it perplexed them. All you actually have to do, you could take her blood, bring it to my lab, put it onto mouse brain sections, and it actually immediately labels all the synapses. It's actually amazing. That's how they figured this out. And so there's actually some easier diagnostic ways. And I think eventually, like many complex diseases, we need better diagnostic tools to know what the underlying mechanism is so that we can actually treat more specifically a very specific mechanism of action.
Okay, time for one last question. Go ahead. Would you
I have a question about hot tub.
Head above the water.
Okay.
So,
Is that, are chemicals used in that typically? Or
Yes.
Is there a
Yeah.
danger?
as long as the water is chlorinated, then you're okay. So don't ever complain when you put your head under the water at a pool or a hot tub and your eyes hurt because it's chlorinated, because it's better that your eyes hurt for a few minutes than to have PAM. But as long as chlorine is in there, then that parasite can't survive.
alcohol would do the same thing.
Wait, I don't. Alcohol in the water?
Okay, so Mike is going to have some brains. I don't think they have parasites. I think they've all been fixed in glutaraldehyde and formaldehyde. And please join me again in thanking Dr. Fox for a very informative talk.